This is the this is the first time I think I've had an introduction where I can say, like, you heard the whole talk, basically, because that's basically what I was going to talk about. So you stole my thunder a little bit with that nice introduction. So thank you for giving it. But I think the purpose, as was pointed out initially, of these lectures is to give a bit more of a perspective on not just the science, but sort of how you got to where you got. And I think that this is an important aspect in terms of the training that you're getting now to kind of sort of see how scientists get where we are, because we all have very, very different stories. And hopefully, as you hear a number of these over the course of the year, some things will resonate with you that you can use to take forward. So the title of my talk is going to be about connecting a passion that I have for chemistry with sort of what I'm doing now, which is sort of thinking about new medicines. And so please keep any questions that you have related to science or my life journey, put them in the chat or keep them. Hopefully we have a few minutes at the end to discuss some things and questions related to some of the things that I'm going to talk about. So in thinking about this talk, again, it's sort of going back in terms of like, well, what led you here, and sort of gives you a chance to be a little bit more, have a bit more of a broad perspective on your life. And so, you can sort of sit back and look at all the things that you've done up to wherever you're at. We all can do that, and sort of gives you an opportunity to see sort of how, does that trajectory make sense, or does it look, like, wow, how did that happen? And so when I look at my career path in terms of sort of where I started and what I'm doing now, I think it looks like this, actually. So sort of stepping back through all of those individual parts in it, it can somewhat look very entropic or somewhat stochastic, because one thing led to another thing, and you can't, sometimes you sort of, there was just a sliver of an event that sort of led to this big change in my life or whatever, but all of the things were very pleasing to me, and I learned a lot from each one of these things, certainly the ones that I selected to talk about today. And then, you can sort of look at your life and say, well, that looks kind of all over the place. And then you think about it a little bit more, and you do begin to find some themes in there, or some parts of it that kind of connect from one thing to another. And in fact, if I were to look through my life, I think chemistry would be a big time thing that's always kind of guided me in terms of the very early days, in terms of where I am now doing what I do, even though I don't think I would have ever envisioned that many years ago. And so, you think about it from that perspective, then life looks a little bit more like it makes a little bit more sense. Let's just put it that way. And so I think that some of us, or all of us, sort of lie on the continuum somewhere between the top figure and the bottom figure. And it's sort of good to be able to understand things that sort of, fit or track throughout life. So I'm going to talk about chemistry today, and I hope that it'll be useful in terms of some of the things that you guys are thinking about, especially the trainees in the audience. So I'm going to start way back, not all the way back, but, pretty far back. And just tell you sort of that growing up, I, really loved science. I can tell you now that it's still, or as early as I can remember, I wasn't reading like, mystery books or kids books. As soon as I learned to read, I was very interested in things, that had to do with science, particularly animals and the human body. And my parents got me a copy of Gray's Anatomy, something that you can find in any bookstore or whatever. And I actually remember that quite a bit. I would just stare at it for long periods of time, and that sort of characterized me. And even now, friends that I'm connecting with way back, or from way back long ago, remember that, like, through the elementary and the middle school years, that I was kind of a science geek. And so that part of it makes sense. So I had a love for science. And then I think there's this whole thing that if you have an inclination to do science at a very early age, you're pretty much, not all the time, but in some corners of life, you're kind of promoted into becoming an MD or medical doctor, because I think for many, that's kind of what most people can associate with a career, that people who are good at science or like to do science end up doing. And so certainly I said, that's kind of cool. And so, I like studying human anatomy and things of that nature. So that seemed pretty much like a logical way to go. Then I went to high school, and my love for science basically led me to do really well in all my science courses, the physics, the biology. But then I got to chemistry, I believe it was in 11th grade. And that was really where I sort of fell in love with a subject. And what I mean by that is, whereas I liked all of the other ones, I think chemistry was more along the lines of maybe reading a novel. I remember just sort of reading my chemistry textbook kind of like it was a newspaper. It was just like a logical thing for me to read, and I picked it up very easily, and it was just fascinating. It all seemed to make sense to me. So I really, really love chemistry. So science is here. Chemistry initially flew way above that. And I learned that in 11th grade. But what do you do with that? You learn that you love chemistry. You still want to become a medical doctor. That's just what you do with science, okay? So I went to college, as one should do, but sort of in the spirit of, well, I like science. Science lover should basically do medicine. And so if you go to college, when you go to college, rather, you have to certainly major in something. And so there's this choice, even though, long-term ambition is to go to medical school, you have to major in some form of a subject. And I didn't necessarily know, to be honest with you, which one I wanted to major in, because it was sort of along the lines of, well, which one's the best one for medical school? So one of the interesting attributes about Howard University, where I went to undergrad, is everybody in their first week in orientation in college had to attend what was called a major fair. And a major fair is kind of like a job fair, if any of you have been to that. So in the job fair, lots of companies there, and you can go to each one and find out about the company and whether they have positions that meet your criteria. In a major fair, you go around to each of the individual departments and you ask them, what are the requirements for the major, so forth, so on. And each booth, so to speak, would have a person representing that particular department. And so I said, well, maybe I should go around the chemistry one. And so this person was standing at the chemistry booth. Name is Dr. Jesse Nicholson. He was the chair of Howard University's chemistry department at the time. And this is one of those moments, and I tell this story, and I never get tired of telling this story because it's so true. It is one of the paradigm shifting moments of my life. And so when I got to the chemistry booth, Dr. Nicholson asked me, he says, what's your name? And I told him, name's Damian Young or whatever. And he says, Mr. Young, what is it that you want to be? And so I said to him what I said to everybody. And, throughout life, when you say, I'm going to be a medical doctor, that's typically viewed as a big time thing. And you get a lot of praise and people say, oh, you must be super smart and all of this stuff. It's really a feel good moment. But he didn't say that. And this is what he said. And I'll never, ever, ever forget that. He said to me this, he said, Mr. Young, would you rather be a person that prescribes a medicine or a person who invents one? And this is the reason why this is very big time for me is because I didn't know what he was talking about, to be honest with you. So, again, I'm looking for the praise and like, oh, that's great. And then he says to me, would you like to be a person that invents a drug? So something hit me at that moment. Number one, it hit me that I actually had never thought of where drugs came from, right? So we all go to the doctor and we get sick, and maybe we get a prescription and we get it filled and we take it, and hopefully our ailment goes away over time. And then we do that again. But we never really stop and think, where do these things come from? And that was my first moment. That was the first time in my life where I actually sat back and said, I have no clue about this. But the interesting thing is, and this is why it was a bit paradigm shifting to me, because even though I didn't know what it was, and all of a sudden now I became very interested to know what it was and how it was done, he seemed to be suggesting that I could do that. And that was why this was such a really cool moment for me. So I'm going to come back to this later in the context of this talk, but I want to sort of start you there. Now, it turns out that Dr. Nicholson would end up being my organic chemistry professor. And little did I know that he was very well known for being the anti-medical school organic chemistry professor. So this would be the guy you don't take if you want to have a chance of getting into medical school. He's very tough. I mean, and you know, that was reflected, I should say, in the enrollment. This course was taught from 4 to 5, Monday, Wednesday, Friday. There were like 12 of us. And the other professors at Howard had like 200 people enrollment. And so, you know, so there were 12 of us and there were like a gazillion other people taking the same class at Howard. And Dr. Nicholson was extremely thorough. He would call on you and, you know, but I love this, you know, and if you didn't know what you were talking about, he would call you out. He would be really like, you know, why don't you know this? And you know, I remember the days of studying for organic chemistry as being some of the most amazing days of my life in terms of learning things, because it was a very new science to me. I'd never understood or never seen this science that involved all of these structural drawings. But one of the things that really resonated with me is this whole reaction mechanism part. And I just thought that was fascinating because, you know, it revolved around the fact that if you learned how things react, some general principles, then you really didn't need to memorize a whole bunch of stuff. And my colleagues at the time who were taking chemistry were like, how do you remember all this stuff? To me, it was very easy because you learned about 5 principles, and those 5 principles kind of came up over and over and over again. Now we call them different names and things like that, but for the most part, electrons and protons like to come together. So that's a general theme in life and chemistry. Pretty much it operates in chemistry all the time. So I love that quite a bit, and organic chemistry was my newfound passion. And so while I was at Howard, I had the great opportunity to do a number of internships, I think starting in my sophomore year. And the molecule on the right is one of the ones that was sort of the center of one of my internships at a company called GD Cyril and Company. So oxytocin is a hormone that basically is excreted in women at the time that it's time for baby to be born. It causes contractions, and the baby is actually forced out of the uterine canal. And ultimately, this is a source of preterm births, because sometimes women go into labor early and you don't want that to happen because the baby is not ready to be delivered ultimately. And so you would like to be able to stop these contractions from occurring. Here's the science here. We're not going to go in that, but other than to say that oxytocin, one of the things that a lot of people wanted to do was to design a compound that prohibited oxytocin from binding to the receptor to start these contractions. And I was able to work on a compound that did that. And I synthesized about 20 compounds during my first summer. And I was able to see that those compounds were indeed active at blocking oxytocin. Now, of course, those never made it into a drug, but it definitely planted the seed for the first time that chemistry was where these drugs came from. So now I'm able to connect what Dr. Nicholson was saying. Would you want to be a person that did that? Now I have actually experienced a research opportunity, or had one, that allowed me to sort of see how that's done using this new thing that I love, organic chemistry. And so that was really important. But what do? You stick to the plan. You're still going to become a medical doctor because you don't quite know how to change your whole mind frame in terms of what you're going to do. So what did I end up doing? I did get into medical school, but to be honest with you, somehow something was missing, in a sense of, you know, I wasn't very excited the day I started getting acceptances into medical school. And as they came in, I just sort of said, oh, another one. And, I got to a point where I said, maybe I'm just burned out, right? So maybe I should take some time off. And so that's what I did. I accepted an offer to a very good medical school, but I decided that I would defer for a year. And I wrote to them and they said, okay, that's fine. We'll admit you, or you can come the following year. And my plan was, maybe I'll just sort of spend the year and just take a break from MCATs and books and all of that, and I'll get a job, I'll earn some money, but I will do something that I like. And so get a job doing chemistry. And so I did. I got a job at a company called Trimeris Incorporated. So leaving DC, going to Durham, North Carolina. And Trimeris was making an anti-HIV drug. And I should say, let me stop for a second and say I got 2 job offers at this point in time, which I thought was really cool, coming right out of college. I was like, wow, I'm in demand, right? But the 2 offers were very different. One was a super mega company that we all know very, very well. As a matter of fact, I think I'm going to talk about them a little bit later in this talk. And the other one was a little bitty company that no one had heard of called Trimeris down in Durham, North Carolina. Why did I choose the one in Durham, North Carolina? Because they're doing something super exciting, I thought, and they're working in the area of HIV. So let me just say a little bit about HIV. Here's a schematic of the virus. And HIV is a retrovirus. It means it basically is an RNA retrovirus. And if you look at the surface of the virus itself, the viral capsid, what you'll notice is that it's studded with these proteins. They're glycoproteins, which means they're proteins that have a bunch of sugars on the surface of it. And these are very important, as we'll see in just a second, called GP120. That's what it is. And buried underneath that GP120 is GP41. I show this because it's a lot of people wonder, well, why don't we have vaccines for HIV? One of the reasons is because of this protein, this virus is highly protected, protecting its proteins with these sugars that are on the surface. There's not a lot of protein in order for your antibodies to really cease. It's really hard for actually the immune system to target HIV because of this. It's sort of it's protected in terms of these GP120s. But there's another reason that this GP120 is important. It's important because it helps the virus to fuse with the T cell, which is the very first part of the virus infection cycle. And so we don't have too much time to go through this, but there are 2 important things that I want you to remember. First of all, GP120 interacts with specific molecules on the surface of your T cell. One of them is a molecule called CD4, and the other one is called CCR5. So when the virus approaches a T cell, it actually sees these 2 molecules, right? And a lot of things happen. This is a very oversimplified version of it. But what happens is this GP120 that I was just talking about actually sheds away when that happens. So CCR5, CD4 binds to GP120, and it actually releases it from the surface of the virus. And what happens is GP120 is actually bound, and it's sort of shielding off this protein called GP41. And when GP41 gets sort of liberated away from GP120, then what happens is it gets very unstable, it collapses onto itself, and it basically pulls the virus into the T cell. And that's really the first part of the virus actually being able to infect a human T cell. And so basically, here's another look at that. Here's what happens when GP41 actually collapses on itself. It forms this nice, what we call 6-helix bundle. It's a very stable thing, and it basically pulls the virus very close to it, and it fuses with the T cell. Now, some very clever folks at Duke, professors at Duke, said, what if you design a compound that actually looks a lot like GP120, the thing that's actually shielding GP41 before this sort of thing happens? Then if you had a compound like that, then once the GP120 goes away, the compound would bind, and it would continue to stabilize GP41 in a conformation where it doesn't collapse on itself, right? And so this was a hypothesis they did and said, well, what would do that? Well, you would design a compound that looks exactly like or very close to GP120. Why don't you use the part of GP120 that actually interacts with GP41? And that's what they did. And so shown here is a 36 amino acid peptide named T20, or enfuviratide. That was the marketed name of this drug. But T20 was in development at the time that I came to Trimeris. And this was really cool because this compound had gone all the way into some early clinical trials, and it was taking people who had viral loads just, through the roof down to undetectable in a matter of a week or so. And people had not seen this level of activity from a new HIV drug. So this is a very new mechanism for treating HIV, prevent the virus from fusing with the T-cell. Now, there's a problem with this. The problem with this is you look at this peptide, and it is indeed a peptide, but there are two parts of this peptide that I'm going to circle here that are quite unique. First of all, this is going from the amino terminus all the way to the carboxy terminus, sort of wrapping around. There are two modifications that are not quite natural. The first is on the amino terminus. It has an acetyl group here. And on the carboxy terminus, it has a primary amide. So this should be a carboxylic acid. It's not, it's a primary amide. It turns out that those modifications are absolutely critical because this thing does not behave very well if you don't make those modifications. Now, why is this important? This means that you cannot express T20 in a normal system like in bacteria. So you had to make it synthetically. And that was the thing that I found to be very interesting. They were going to make this synthetic compound, or they had already made it actually by the time I got there, to be administered as a drug. Now, if you look at all the various steps involved in connecting amino acids and things like that, don't have time to show that, 106 steps. And that today is still the longest synthesis of any FDA-approved drug that is on the market or has ever been on the market. Certainly, this compound is not prescribed anymore. There's certainly much better HIV drugs. But at the time, it would take 106 steps, and they needed very effective chemistry to be able to do that. And I was part of a team that actually helped. One of the key steps that I actually was responsible for increasing the yield about 5% of one of the critical steps in the synthesis of this molecule. And that, if you did the calculations on batch, that saved about $180,000 per batch just to make that increase in yield. And it actually made the synthesis a lot more palpable. So I was able to actually see the power of chemistry in terms of my ability to actually go into a hood and do some reactions, optimize some reactions, and then actually that gets translated into a compound that eventually became FDA approved. And so I was really hooked in this 1 year off to do chemistry. I really sort of saw the power. I was hooked, and I wanted to learn more. So, toward the end of that year, I sort of did a self-analysis. And so this is sort of one of the key, again, things that people do. Sometimes you get it from someone else, sometimes you get it from yourself. So I sort of began to learn some things about myself. Number one, I came clean with myself and realized I didn't quite like to be around sick people. They didn't feel extremely comfortable. Medical doctors are amazing people, and part of it is because they can do that day in and day out. And I don't like hospitals, so these are things that just didn't speak to me. And so I was clear about that. The second thing, I like being in the lab. So that was really something that I thought was interesting, what I didn't like, and then what I liked. The third thing, I was okay with not knowing answers, and I actually didn't mind sort of the chipping away at it. And I think, I think my personality is more toward that. I actually like that sort of puzzle aspect of life. Whereas I think if you're a doctor, the puzzle needs to be solved very quickly. It's certainly much more advantageous for you to know right away what's wrong with your patient, rather than sort of, pontificating about it. They want to get well now. So, you tend to need to sort of get with it. Whereas, I sort of like problems that kind of you sit with for a very long time in sort of a mountain climbing exercise. All right, I did not like the fact that, up to that point in time, certainly knew that there are gaps in medicine where you have a loved one that is sick and there's not much that medical science can do for you. So I had, a father and two grandparents that died of cancers that were really so aggressive, and there really weren't any treatment options whatsoever for them at the time. And so that was something that I was very keen to, that we need to do better. We need better types of medicines to be treating people. And then finally, I did love chemistry. So I came back to that. And that year off really gave me the ability to sort of say, this is what you really ought to be doing. So we didn't want to become a medical doctor. Now I'm going to become a chemist. And so I was able to do that self-analysis and then come to the conclusion that I think I needed to sort of follow my heart and my passion. And so that's the bottom line of this talk. And, you know, if you have something else to do, you can certainly go, because I think that's something that's the major point of what I want to say. The rest is sort of downhill, I think. So I went to graduate school, and I did what people do in graduate school. I think people are doing certainly a lot more things in chemistry now, but at the time, there were two major activities in chemistry if you wanted a PhD. Number one, you synthesized a natural product, which is a compound of usually extraordinary complexity that nature has made. And then the question is, well, chemist, are you able to reproduce that in the lab? And so that's a major intellectual exercise. The other one is to develop new reactions and things like that. So those used to be the two. I chose the first one. I was really kind of infatuated with these molecules that come from nature. And so here's the molecule that I worked on in graduate school. It's called spirolucidine. And as you can see, it has a lot of features that are quite interesting in a sense that it has these rings. They're connected in various ways. It has this ring right here that's called a spirocyclic center because it's part of two rings. And that's what it derives its name, spirolucidine. And it comes from a club moss. So this is sort of, I put this in the parentheses. This is sort of what it comes down to here. You synthesize that, you get a PhD. And as I came to the end of my graduate career, it became clear to me that, you know, that while I enjoy chemistry from the perspective of all of the great things that you could do, and I certainly think I became a very proficient chemist at sort of doing this, and that's one of the reasons why this activity became very popular, doing synthesis of natural products. But I found that I really searched for more in terms of what you could do with this chemistry. I'd come off again the high of seeing how chemistry could impact people with HIV who didn't have very good drugs, or the drugs that they were on were failing. And then all of a sudden those drugs, the drugs that I was able to provide with my own synthetic know-how, was helping to generate a better situation. I wanted to connect more to that. So in the end, I thought that I learned a lot from graduate school. I certainly love thinking about chemistry. I learned a lot of reactions and mechanisms like we were talking about earlier, but I really wanted to solve more relevant problems. And it was quite interesting because it was somewhat really interesting that I got offered a job to be a professor the day I turned in my thesis. And so that was a very cool thing, because like, wow, I don't have to actually, you know, I would have a real job, right? One that, you know, was like a career job, but I didn't know how to do what I thought I wanted to do at that time. And so I said, I have to do a postdoc. I have to learn more. And so that led me to the next great place in my life, which is Cambridge, Massachusetts, to Harvard University. And you know, one of the things I can say, I always say to people, you know, Boston is a fantastic place. Harvard's a great place too. But, you know, just being in Boston, which is really a hub for biotechnology and medicine, is an extraordinary experience no matter where you end up. And I always tell people at some point in time in your life, you should go through there, whether it's undergrad or, you know, toward the end of your career. It's a great place to be. So what did I do in Boston? So again, I became very sort of like, well, what else is there to do with chemistry? I know how to make molecules. It became clear there are two types of molecules at the time. Molecules that look more like drugs that chemists had been making for 30, 40, 50 years. And then on the other end, I've already introduced you to those natural products. So Taxol, which is an anti-cancer natural product, is an example of that. Now you can see that the structures of Taxol, the structure of Taxol rather, and the structure of the molecule on the right, which is Valium, it's a class of drugs called benzodiazepines. They look very different. So on the left, you have this two-ring structure that mostly, if I were to draw it out in 3 dimensions, looks fairly flat. Okay, so most of that structure could sort of sit, you know, on a table. On the right-hand side, you have a structure like Taxol that, if I were to put it on the table, it would be very three-dimensional. Almost we could call it spherical. And so, structurally, these things look very different. And in fact, their activities are very different. They act very different. So the types of things that chemists make that act on targets tend to be very different from the types of things that nature makes. However, if you can sort of see the molecule on the left as being what is the level of difficulty to make it, you might say not very compared to the one on the right. And you would be right. It would take far fewer steps. As a matter of fact, you can make Valium in 2 chemical transformations, probably give it to anybody who's had their undergraduate chemistry lab, and they could actually make Valium. I don't recommend doing that. The DEA does not allow you to do that. But you could do it if I gave you the right things. Now on the right side, the Taxol, it would take you probably a minimum of about 30, 40 or so transformations, okay? Now, what if there were another kind of molecule, a molecule like the one I'm showing in the box, and it comes about from a process called diversity-oriented synthesis. Now, what you would sort of look at, if you look at this one, you might say, it has some features of the Taxol one. It doesn't look quite as complicated as that. But it looks way more different than the Valium molecule. It has these so-called stereogenic carbon atoms that I'm representing in stereospace by the bold and the dash bonds. So things coming out, things going back. So it is more three-dimensional. And so the question is if you have an intermediate number or intermediate amount of complexity, would that correlate to an intermediate amount of synthesis? And it turns out that it does. And so I learned how to do this in my postdoctoral fellowship. And I think I was pretty good at it. At the time, my postdoctoral advisor, whose name is Stuart Schreiber, was starting an institute called the Broad Institute that some of you may have heard of. It's sort of the credited, or one of its biggest credits is sequencing of the human genome. And so ultimately, the Broad basically does a lot of science, or a lot of areas of science related to medicine and genomics, et cetera. And chemistry was a platform that they began to build. And I became one of the first hires sort of on the chemistry side to do this new way of synthesis called diversity-oriented synthesis to generate a very unique compound collection. So just shown here, we're not going to go into the chemistry, but we were able to sort of start with some sort of central structures here and convert these things to a wide variety of end structures that could be tested. So what I'd have you appreciate is the fact that this same intermediate here shown in the center can go into making things medium-sized rings like 8 or 9, all the way up to macrocycles like 14. So all of the structures that I'm showing you at the periphery came from that central molecule, and that was a way of doing the synthesis that we pioneered at the time. And did it work? Yes, it did. Turns out that these molecules, if you screen them against a wide number of disease targets, they provided hits: psychiatric diseases, cancer, diabetes, infectious diseases, et cetera. And so we made about 100,000 compounds in that collection, and they continue to this day to be starting points to many disease-related programs. Even though I'm not there anymore, that science lives on, so that's quite pleasing. So that brings me here. So after spending some time at the Broad, it was time for me to figure out what I wanted to do in terms of my own independent research career, because I was more of a staff scientist after I finished my postdoc, that time between postdoc and here. And so now I decided I wanted to go back more to the academic side of things. And I was recruited here by Dr. Marty Matzuk, who's now the chair of pathology and director of Center for Drug Discovery. Thank you, Marty, if you're in the audience. And so I came here to begin thinking about ways of doing drug discovery in a way that we can sort of impact on some of the key challenges. So basically, I think it's one of scientists' responsibilities. We certainly, I've used this slide for many years now, actually, and now I think there's no truer time to sort of think about this than now, is that one of our responsibilities as scientists is to understand disease and help keep the public safe. But however, one of the unfortunate observations is that sort of we think about how drugs are made over the last 20 years. we've made a lot of impacts in a lot of areas, but our drug discovery, the generation of new drugs to treat diseases hasn't really kept up. And so that's unfortunate. And it's sort of compared, it's a very different way of thinking about that than sort of other areas of science. And I'm showing here computers. So Moore's law, basically, Gordon Moore worked for IBM, and he basically hypothesized that, basically if you go further in time, computer technology would get smaller and its ability to process information would get better. And so ultimately that is held up true. Gordon Moore was very correct in terms of computing hardware. But if you were to sort of apply, well, is that how that works with every area of science? It does not. And drug discovery is one of those areas. And if you sort of look at drug discovery, these scientists wrote a very interesting article back in 2012 that called it Eroom's laws. If you spell Moore backwards, you get Eroom. And it's exactly what you have here is the inverse of Moore's law. So instead of going up, as we go forward in time, drugs are going down. So basically, this is a quote out of that paper, and the number of FDA approved drugs has halved every 9 years since 1950. And so now, if you continue that arc out a little bit more, you would see some blips going up and down. But, for the most part, the way that I would say is that we can do a lot better than we are. And there are a number of reasons why we don't do as well. And, we don't have time to go through all of those in this talk, but I'm going to talk about a few things that I think we can do. So this is just looking at some statistics related to drug discovery. Drugs get ultimately developed in the lab, and then they go into clinical testing and all the way through phase 1, which is generally the drug is not given just to see to sick people. It's given to healthy people to determine toxicity. And then phase 2 is actually given to a small cohort of people who have a disease. And then in phase 3, a much larger one. And then after that, if all that data looks good, you're able to apply for something called an NDA, which is a new drug application. Now, if you look at the success rate from drug discovery, this is looking at academic drug discovery, by the way. Now, if you look at the things that made it prior to getting to the clinic up to phase 1, 798 projects. 254 of those made it into a phase 1 clinical trial. So you got a lot of attrition there, so about 23% success rate. But then once you enter that, there's only about a 9% success rate of a compound that would make it into FDA approval. So combined, that's about 2% success rate. 2 drugs out of every 100 that would get made would actually drug programs rather would make it all the way to FDA approval. And so that's abysmally low, right? I think we would all agree with that. So just a few other numbers in addition to the fact that, drug discovery has a low success rate. It's very expensive. So $3 billion is about the average program now. Certainly they can go beyond 5 depending on the disease area. We're still taking somewhere between 12 and 15 years to advance a program from the bench all the way to FDA approval. I just talked about the high failure rate. And then going back to what I was talking about earlier, certainly in the case of my family, there were no therapeutics to treat the cancers that they have. And so we always have to be thinking about, there's clearly a way that we can do this better. So some people would be quite surprised to find out that one of the reasons why drugs fail so prevalently is not because they have bad or toxic effects in humans. The fact is, shown here, efficacy versus placebo. These things sort of fall out in phase 3 clinical trials. So they've been proven to be, safe and things of that nature. They fall out because they don't do anything. They actually don't have efficacy at all. And that is a big problem that we are trying to solve because ultimately we think we understand the disease very well. And we say, if we can design a compound that will target this thing or modulate this protein's function, it should behave as a good drug. And a lot of times, or most of the time rather, we are wrong, right? So we need better ways of figuring that out. So here are the 3 things that I would like to sort of begin wrapping up with in terms of what am I working on now? How does chemistry impact on some of the things that I just said in terms of the failure of drugs to sort of progress to patients who need them? So one of the first challenges is of course, the target. We talked about that. If you pick the wrong target, you'll get a compound, it'll inhibit the protein's function, let's say, but it won't necessarily work in humans. And this is a problem. This is why most of the drugs fail. After you find a target, let's say you got a really good target, how do you find a starting point, Well, we typically screen. We don't have there's a lot of really exciting science, and I'm really looking forward to sort of machine learning and AI coming into play over the next 5 years to 10 years in drug discovery. But at this point, the best we can do is still to screen in one form or another. There are a lot of ways of screening, but that's what we do to find a starting point for our target that we've chosen. Not all screens are the same, and they have their issues. The last thing is sort of, I would say, is can we do this faster? I mentioned something about 12 to 15 years earlier. That is a lifetime, especially I think we've been faced with, since we've been faced with COVID, to think about that time scale when people are dying in real time. And so that is just unacceptable. We have to do better than that. So I'm going to just give a little vignette on each one of these. And first is picking better targets. And so, back when I was in learning about the brain, it used to be sort of thought about that, you're born with all the neurons that you're ever going to have. So good luck with what you have. And, that's somewhat true in a sense of fully differentiated neurons in the brain. They don't replicate. But there is a part of the brain called the hippocampus where some really interesting stuff happens. And that is, there's a population of cells called neural stem cells. And in fact, those neural stem cells actually, they self-create themselves. So they replicate and they form new neural stem cells, but they also have the ability to differentiate into full-blown neurons through the process I'm shown here. And so it turns out that a healthy human, as you grow, you produce about 800 to 1,000 new neurons per day. And that process continues throughout your life unless things go wrong. So we know that to be the case, but we also know that in diseases like Alzheimer's disease and depression, major depressive disorder, this process of neurogenesis doesn't occur to the same extent. And so basically one might hypothesize that if you can boost this hippocampal neurogenesis, then you might have an effective treatment for diseases like Alzheimer's or major depressive disorder. And so no time to go into this, but there is a lot of biology known about a protein called TLX. TLX is what we call a nuclear receptor, which means it's a transcription factor that gets activated by ligands. That's good. Ligands are typically small molecules. And if you can activate TLX with small molecules to do neurogenesis, that could be a good thing. So TLX is involved in lots of processes that we would like to be able to control. And that's something that, we decided to look into. A research collaborator and I, Dr. Mariana Mladic-Savadic. Now, her lab had already established a number of interesting things about that ligands that could potentially bind to TLX, but they weren't known. So a lot of nuclear receptors, or you have the known, so basically the estrogen receptor is a very famous one. It binds estradiol. TLX is a nuclear receptor, but it didn't have any known ligands. And so that would be useful to figuring out if you wanted to drug it, to activate it, to cause neurogenesis. So our first question was, can we figure out what those natural molecules are? And over a course of a number of years, we figured out that in fact, it's oleic acid, and oleic acid is the most prevalent ingredient in olive oil. So you might immediately say, well, that's fantastic. I'll just go and eat a bunch of Italian food or some good Italian food, and I should be good to go on my neurogenesis. But it doesn't work that way. It turns out that this oleic acid, or cis-18-1 omega 9, that's another name for that, is made in the brain. So the body has a way, in the brain, there's a way in which it actually makes this specific fatty acid, and then that triggers TLX to cause neurogenesis. So oleic acid isn't a very good drug, no matter how much, or if you drink olive oil for breakfast, let's say in the morning, which I don't encourage one to do, it's probably not going to impact your neurogenesis that much. So the question is, can we then make molecules that do that? And in fact, the answer is yes. we already have. We've been at this for a number of years now. We made molecules in the lab that actually can bind to TLX and cause neurogenesis to take place. And so the idea is that, you know, TLX agonists, we call them, they will cause these neural stem cells to proliferate, and then they will also cause them to differentiate into full-blown neurons. And so we're very excited to begin testing these. Actually, we're doing this right now as we speak, testing this in a mouse model of Alzheimer's to determine whether the hypothesis is correct, if increasing neurogenesis will decrease the cognitive decline of Alzheimer's patients. Okay, innovation number 2 is, can we find better starting points? This comes down to a challenge of chemical diversity, and I think this is a very important concept to think about. I'm not going to go through this, but typically we screen for molecules. The vast majority of pharmaceutical companies would screen about 500,000 to 1 million compounds to try to find a starting point. And they would do that in these individual wells. And some of you may be very familiar with this. But the idea is, if you have a target, you screen compounds in these wells and you would hopefully find a hit. But there are a number of problems with that. And major problem is the fact that the compounds that you screen must be chosen correctly. Otherwise, you may not get a good starting point. And this is a good study from GSK that sort of demonstrated in their ability to go after antibacterial targets. They screen about 70 targets that are involved in microbial biogenesis. And these were defined as essential. So these would be good drug targets if you can design a molecule and knock them out, right, to hit them, to inhibit their function. So shown here are those 70 targets that involved in various aspects of the bacterial life cycle. And then they screened, doing that high throughput screening I talked about, and they got basically nothing out of every single target that they screened. 500,000 compounds per screen, and this is just one page of the many pages in this paper that sort of give you the gross failure that they had to produce anything out of that. And what they said at the end is that chemical diversity is not quite ready. That's what they said at this time. This was basically done in the late 90s, and this was a concept that was starting to come on board. And so, you know, I like to compare high throughput screening collections to this. And you look at these, red, yellow, green, they're all apples at the end of the day. But this is what you need to target a wide range of disease targets so that you would be able to find one starting point. Once a chemist can find a starting point, then they're able to optimize that. But you have to find a hit. And in order to find a hit, you probably need a lot of different types of molecules with a lot of different structures, similar to the fruits shown here, not just red apples and green apples. So we are doing something called fragment-based drug discovery. One of our approaches is to do that. Fragments are quite smaller than normal drugs. Normal drugs are about 500 molecular weight. Fragments are about 250 or smaller. They typically bind very weakly to targets, so about 1,000 times weaker, but still you can optimize them to very potent hits. And this has been shown over and over, and this is another parallel way of screening. So you can grow the fragment to make it a better binder. Sometimes you can link them together, but both of these techniques are well demonstrated in the literature and in the pharmaceutical industry. Now, going back to the thought of, well, what have people used for fragments? Well, before we got into this sort of arena, people were sort of using the red apple, green apple approach. So similar to the GSK study, the diversity of those fragment collections was not very high. And so what we did when I came to Baylor is to initiate a program aimed at looking at fragment structure to diversify them. So shown here is it doesn't matter what the name of it is. It's this triangular plot that sort of depicts a way of viewing molecular shapes. So linear is up in this corner, well, disc-like, sort of flat in 2 dimensions in this corner, and then disc-like or spherical in this corner, spherical in this corner. And if you look at what we've done, we designed chemistry to create these smattering of dots that you see. There are 3 different ways that we did that. And it sort of distributes this in this PMI, which is called principal moments of inertia space. Most compounds that pharmaceutical companies screen lie along this flat edge here. And this is why we believe that there are not hits to a lot of targets. Some they do very well against. Not all things require things that are more 3D like, but a lot of things they don't. And we've been able to apply our compounds to TLX, and we have shown that we can take our fragments and we can target TLX. In this case, we can activate it. That's what this is shown in this assay called an alpha assay. It's mirror image compound, which is the right hand, left hand. If you take the right hand, it's a hit. The left hand is not, and that shows the specificity of our interaction. So this is a really important aspect of what we've done to sort of diversify the types of structures that are needed to do fragment-based drug discovery so that we can target a wide variety of targets. Last but not least is, can we deliver therapeutics more rapidly? I don't have to introduce COVID-19. That's why we're not sitting in person now. But ultimately, it's impacted all of our lives in a way that we can't begin to imagine. The vaccines, thankfully, have allowed us to come back to some normal semblance of life. But as we all know, there are issues with vaccines, including the fact that there's a lot of public skepticism about them, but also the fact that there's the emergence of viral variants that may cause the virus to escape the immunity that we build up to the vaccines that we've already taken. And some of you may know already that the FDA will not be approving boosters at this point in time. That came just about a couple hours ago, about an hour before I got on the talk. So we're always going to have an issue with vaccines. And I think that at the end of the day, and this was noted by the director of the NIH, we need drugs as well. So you're going to always need the ability to prevent infection, but also if people become infected, you're going to need to have antiviral drugs that actually will allow their bodies to rid themselves of the virus. So we've been using a technology called DNA encoded chemistry technology, which won't have a chance to go into what it is, but it allows for billions of compounds to be screened that have a DNA barcode attached to it. So at the end of the day, each drug-like compound, each of the billions of different drug-like compounds has a unique sequence of DNA that corresponds to this. And that's quite useful because if you recall, that in the high throughput screening, we had to have those individual wells where each compound goes in a well. With this one, you don't. You can screen in mixtures. And so this is a really cool way of screening. You take a library of DNA encoded compounds and you have a protein of interest, you can actually tag it with what we call an epitope tag. That allows you to incubate the protein with the entire library, so all billions of compounds. Some compounds will bind, some won't. You can use the tag to actually pull the protein out of solution along with the things that are bound, the compounds that are bound to it, wash away the non-bound compounds. And that's how you isolate them. Then you can actually sequence the DNA through a process that involves amplifying the DNA barcode through PCR and then sequencing the DNA. That gives you the identity of the compound that bound to your protein target, which you can then go back in and then resynthesize in the lab, traditional way of doing that. Didn't tell you how this is done, but it's done in a very different way than how we typically make molecules. And then you can do structure confirmation and test on to determine if it's a good drug. So just to wrap up, we did this with Impro less than a year ago. We took a protein that's involved in the coronavirus life cycle, called its main protease, which has a specific function in the viral life cycle. And we did this DNA encoded chemistry screen because it's a very efficient way of screening. And right away, we came out with a hit that we were able to make off of DNA and test in a number of tests. And in fact, not only does it inhibit the main protease, but it does so very well. It's a highly potent compound. And we've even gone forward to show that we can crystallize it and show exactly what the binding details are and things like that. So if we needed to optimize it, we could based on this picture that we're seeing now, that the compound was very potent coming right out of this DNA barcoded screen. But most importantly, and this is what I'll leave with, is the fact that this process was very quick. And so from start to finish, I'm just showing the major aspects that are involved in the screen, from getting the protein to doing that DNA encoded selection, to doing all the stuff that we would do to make the compound and then test it, obviously do the antiviral testing, took a period of less than 3 months. And so this is very exciting to us because this will speak to not only the current pandemic, but unfortunately pandemics maybe that could be coming in the future, or maybe even just this same one, being able to respond to it with drugs in a very fast and rapid manner. So 1 minute on diversity. This is a talk about diversity, and I've talked about it in the perspective of compounds, but diversity matters certainly in terms of people. I firmly have benefited from always being a part of teams that were formed from people that came from all walks of life and different perspectives. And that has made for me to always be excited about staying in science. People think of science as like you're this person that goes in this lab, and you're crazy and, you just sort of talk to yourself. And that's not what we are. We're social beings. We like to interact. We like to eat food. We like to share experiences about life and science. We have different perspectives on that. And, in true form, one of the reasons why I truly believe that I'm here is because of this individual. So I started very early with Dr. Nicholson. It was a mind-blowing thing. The third thing that blew my mind was he was an African-American scientist, and I had never seen that before. It never occurred to me that either at the point in time that I met him early in my college years. But that meant a lot. And the fact is that, maybe I wouldn't be here, or I would be in medical school, or who knows, had it not been for this person. And so diversity matters. And, I think that this is something that I would like to leave you guys with. So here's my career path again, but here's also my career path. I followed chemistry. I tried to, take the passion that I have for it to carry me forward. And I'm really excited. I've always been very excited, and I continue to be, about what the future holds. And so with that, I'd just like to say that these are the scientists. I think this is a fantastic way to end in terms of diversity. A picture of the Center for Drug Discovery and the scientists I'm very fortunate to work with every day, tackling very important problems related to drug discovery, and they make life enjoyable and science that can impact humanity. So with that, I'd like to thank you, certainly for sticking with me on a Friday afternoon when you probably have a lot of other more fun things to do. But thanks to the organizers for inviting me. Yvonne, thank you for the introduction. And thank you all. Bye-bye.
Thank you, Dr. Young. That was a very interesting talk, and also very inspiring to hear your journey as well.
So I don't know what we're doing here. So are we taking questions? I know we're at 5 o'clock now. So
yes, so we're welcoming questions. If someone has them, just please unmute.
Hi, Dr. Young. I have a question regarding the DNA barcoding. So I was curious if the DNA barcode tags were removed after candidate small molecules were selected, and if so, did you see any unexpected or decorrelated effects on their affinity to targets? Like, were some promising candidates lost once you removed the DNA barcodes, or was there any real significant effect there?
Yeah, that's a great question. Yeah, so we do, once we do the sequencing and all of that, that then begins to make it the regular way without any DNA on it. So that's what we do after we get our sequencing analysis and find the binders to it, and we test them. And I would say about 80% of the time we find that the compound that we get an enrichment with that selection actually does bind to it. We don't test the actual affinity of the compound with the DNA barcode because there's just so little of it that we can't do some of the binding assays and things like that. So to truly answer your question, to sort of binding affinity with the barcode versus not, we don't have that comparison necessarily to know what it was with it on, but it does bind with it off usually. And there are some cases where people do have the ability to synthesize things with a barcode on and test them. We don't necessarily do it that way, but in those cases, you would see if it binds a very similar affinity, usually somewhat less in the cases that I've seen. And that's probably just because molecule tumbles a little bit differently through space. It's a bigger molecule, but on you definitely see binding on the same order of magnitude as you would see sort of with the DNA on. And this way of selecting, I should mention, but we do this in multiple rounds. So you typically select for very high affinity binders to your target. So we're typically talking 50 to 100 nanomolar right away, which is like almost unheard of in most regular high throughput screenings. So I hope that answers your question.
Yeah, absolutely. Thanks so much.
Here's a question from the chat. Are there limitations to DEC tech when it comes to chemical diversity of drug-like molecules, or is it improved comparing to HDF?
Oh, that's a very good question. So yes, there are major limitations. So DNA-encoded libraries gives you large numbers of compounds, but the way that we generate those large numbers of compounds is we typically create a sort of central core and we decorate it with lots of different building blocks. And that is diversity to some extent, but is not really diversity in the context of what if you could make with lots of different core things. That's hard to do, just in general. But number two, I'm going to say that I didn't touch on this, but to build these molecules, we have to do it on DNA, obviously, and that means that you have to be limited by certain things. And number one, your reaction can't harm the DNA, nor can the DNA shut down the reaction. So that's not trivial. Number two, solubility. DNA is pretty much only soluble in an aqueous or mostly aqueous environment. And that means that, a lot of organic reactions that we know and love don't work in water quite as well. So that contributes to the number of reactions that we can do. So those two things together make us have to think very creatively in terms of how to generate chemical diversity. But the good news is that there are enough reactions that we can use quite with high fidelity. And we are continuing to innovate in that regard. So if the reaction that we love in the normal organic sense isn't we can't pick it up and do it in dichloromethane, which is a solvent organic chemists like, or THF, then we try to figure out in the center ways of actually taking those reactions we love and making it work on this platform. It takes a whole new rearrangement of your chemical brain to do that. You need some neurogenesis to do that, but you can do it. And so I think that's another cool thing with chemistry that I've loved to do, is sort of think about that, to create more diverse compounds. So at the end of the day, long story short, we want to be able to generate not just large numbers of compounds, but also structurally diverse molecules. And that is an intellectual challenge that we work on quite a bit. Yes, good questions. Anybody else?
Here's thoughts on how impactful synthetic biology will be for drug synthesis.
Synthetic biology. Yeah. For drug synthesis, is that what the question says? Well, yeah, I think certainly some amazing work going on there. And, many scientists who are in the field of drug discovery would maybe say the challenges that I have outlaid aren't the same ones that they would say. So maybe they're thinking like, biology can make more naturally occurring like molecules, or let's say, here's an example, we're sequencing genomes of ancient organisms now, and we're trying to, or some scientists, not me, but are trying to determine what natural products may have existed in the past that don't now, and sort of unearth those. that's kind of a synthetic biology kind of thing. And, that's yielding some very interesting results and may actually impact on drug discovery. So I think that, not my field, but I think that, it brings up the point, your question, in a sense that many types of scientists are needed to sort of impact on the question. Synthetic biology, certainly, I believe some really creative work is going into. So I'm not sure which aspect of synthetic biology you're talking about, but I definitely say very impactful and very creative.
Hi, Damian. I typed my message, but I think it's a lot. We can talk about it later too, but it was about the TLX and oleic acid paper. And I'm interested in lipid transfer mechanisms. And I was wondering if anyone else brought up like the topic of studying the relationship between the liver and the brain in regards to neurogenesis and Alzheimer's studies.
Well, so Peter, since you're in my lab, let's pick that one up on Monday. So that's a great question. Maybe we have a few more general ones that people might want to ask. If not, so I always end with my email because some people are shy and don't like to ask questions, or they'll think of things later that they didn't think about in the context of the talk. So feel free to talk to me or email me, and we can even meet up with our masks on and things like that if we want to have coffee and stuff. So I like talking about career related stuff, and if I can be of any help to any of you, please let me know. And certainly if you guys have targets, I know there's a wide ranging audience. If you guys have targets, please reach out to me as well. We can determine whether or not it would be possible to screen to do some initial drug discovery. So hearing no further questions, thanks for coming and have a great afternoon.
Thanks so much, Damian. We'll see everyone next week with Neil Waxham from UT Health. Thanks for being here and have a great weekend. Bye-bye.
Thank you, Damian.